Okay, so our topic today is multivariate inheritance, and just as we talked about response to selection, we're going to just talk about response to selection when we've got multiple traits. And it's a little non-intuitive, and we'll do a little matrix algebra today as well. Okay, so we have an organism here. This is Wilson's bird of paradise, and I'll talk about birds of paradise later on in the course. We'll fit some alternative models of evolutionary process to the bird of paradise radiation. And I like Wilson's bird of paradise for two reasons. The main reason is that it's clearly a multivariate guy, right? I mean, he's got these bizarre color patches. And if you just think about a normal bird, it's got these crazy plumes coming off the wings, but the color patches are just, this is why this radiation is thought to be the iconic example of radiation bisexual selection. And I also like birds of paradise, Wilson's bird of paradise, because he's got male pattern baldness, and highlighted in blue at that. But the idea is he reminds us that we, in general, we need to think about multiple traits when we think about evolution. Traits they aren't inherited singly, and they aren't acted on by selection as single entities. Okay, so here's our thesis for this first lecture. And that is the statistical approach we were using for the inheritance of a single trait can be extended to multiple traits. And when we do that, instead of just heritability, we're going to find that actually heritability turns out to be not a particularly useful concept in the multivariate case. Instead, we're going to be using something called the G matrix, which is a matrix with genetic variances, those G's on the main diagonal for each trait, and then, as you'll see, genetic covariances in the off-diagonal areas. And then our third statement is that just as heritability affected the response to selection on a single trait, when we look at the response to selection on multiple traits, it's going to be the G matrix that affects those responses. And so when we want to look at the change in the trait over a single generation in response either to selection or to drift, finite sampling of parents, it's going to be this G matrix that's going to be useful to us. Okay, so when we go to the multivariate case, when we look back at the univariate case, we go, oh, well, maybe we shouldn't have been using heritability. Maybe we should have been thinking about G there, not just in the multivariate case. So in general, there are important lessons to be learned from the multivariate case. Okay, whoops, here, let's go here, whoops. Hit the wrong key and you're penalized. Okay, here's the outline of the topics. It's going to be a pretty simple lecture, actually. We're going to, first of all, start and just look at multivariate resemblance between parents and offspring, and as it is captured by the G matrix. We're going to look at a multivariate model of inheritance. We're going to look at some examples, primarily garter snakes, I think. The G matrix is affected by opposing forces, just as G, the genetic variance, can equilibrate under opposing forces in the natural world. It's virtually the same idea in the case of the multivariate G matrix. And the G matrix affects the evolution of the multivariate mean. We'll allude to drift, but we're going to illustrate multivariate, the change in the multivariate mean as it's affected by the G matrix. Okay, so the new concept that we have, yeah, Tyler?
Yeah. The PowerPoint lecture for this is the single trait lecture.
What?
Yeah. I don't have the media, I don't have the multiple traits in the PowerPoint.
We can fix that. Yeah, but I've got, yeah, you don't want that one. Oops. Hang on. No, and I pulled this up not from the website, but from my, I'm always ready for recovery. Whoops. Okay, so you'll see NIMBioS 2015, and you'll see lecture 2.1, and there are two PowerPoints. One has plus links, and that's the one you want to put on the website. Now, sorry, guys. I didn't realize. We should have checked that, but it's funny, this is upside down. All right. Recover. You're not with me then here. Where are you? Thanks for saying something. Or was it Tyler? You all were going to sit quietly by and let me proceed in isolation, huh? I thought we got over your shyness yesterday. All right, but you can watch the screen for now, even if you can't see it on your computer. Okay, so we'll illustrate multiple trait inheritance with two traits, just for simplicity. And the concepts that you get from two traits are sufficient to handle the multivariate trait. The matrix is just bigger. The elements are the same. So when we look at two traits, if you want to think about the expression of heritability in a single trait, it means that the trait is going to run in families, that it's going to be associated with there are going to be differences between families in the trait. And when we think of two traits, then the expression of genetic coupling or genetic correlation is reflecting the fact that the traits are going to run together in families. And that running together in families is the phenomenon that we're going to, the new concept that we're going to deal with today in inheritance. And here's two expressions of that. If we go back to our garter snake data, remember when we were plotting it, we had moms and kids. We plotted kid's body against mom's body. We can do cross-trait plots. And here we're doing one of those. Here's mom's body count on this axis and daughter's tail count on that axis, right? And we see a small, but as we'll see, you're okay, significant slope, non-zero slope. We can also do the reciprocal thing and plot mom's tail count on the x-axis and the average daughter's body count on this axis. And if we do that, we get a slightly different slope. As you recall, a lot of the moms had missing tails because of the slings and arrows of outrageous fortune. They ended up losing tips of their tail, and they get deleted from this data set. And so you don't exactly, you have a different sample size. But theoretically, these two distributions should be estimating the same parameter. Now, what's useful is not going to be the slope, but the covariance of this plot. And so just as this plot, in the heritability sense, estimated 1/2 the genetic variance, here it's estimating 1/2 the genetic covariance between the tail and the body count. Likewise, and this plot should be estimating the same parameter, even though these point estimates look a little different. Okay. Can I move what? Yeah, that's an awkward place to leave that. Good point. All right. We left that up there to elicit audience participation. I'm glad it worked. You want to see an animation? Here's the animated. Whoops. Should work. Let's see what I have to do here. I probably have to click there. Come on. Where's my, here you are. Maybe you're getting this to work. All right. Connect to Wi-Fi. Why didn't you do that automatically? It's an irritating feature. You have that problem too, don't you?
I had to re-log in today too.
Huh. Oh, you had to re-log in? Oh. Are you? Did you get a pop-up window if you're logged into the site? Or did it go like that? Yeah, okay. I won't bother to do it. I just wanted to illustrate. So this animation, as like the others that I'll show, are on this website that I have for animations that illustrate this book I'm writing. So up at the top, you'll see a ribbon on the website, and you can go see, you can find a table of contents there, and you can find information in the book. Is there a ribbon up at the top? You're not giving me a lot of feedback here. You don't have, come on, Dave, jeez. No, isn't anybody looking at this? Yeah, yeah, okay. Yeah, there you go. Good man. So up at the top, you see a ribbon, and it says, move about here. Scroll up to the top there. So that, yeah, there you go. So you can read about me. You can read about the book, the rationale, the tutorial. It's a link to the tutorial. At any rate, you can see, you can go to the website, and then you can get into all of the animations, not just the one in the pop-up window. All right. I didn't realize we had this complication, but... All right. Okay, so the concept of genetic covariance. All right, so let's back up and think about what kind of a model we need for inheritance. And so this is the value of introducing the Landy notation last time, because now it's going to be very simple in the multivariate case. So here's our same symbol for the multivariate, for the phenotypic value of an individual, Z. Here's the breeding value, the sum of additive effects over loci as they affect that trait. And here's the environmental values. Okay, so in the multivariate case, illustrated here with the two-trait case, there's a column vector of two values. So that might be the individual's body vertebral count and the individual's tail vertebral count. And by doing something like, what's our McGLM, the Hadfield software you're using with Pat Carter yesterday, you could actually estimate, if you had enough data, you could estimate the breeding value for body vertebrae and the breeding value for tail vertebrae. And then here's the environmental value, which includes everything, any genetic values that aren't additive genetic, like dominance and epistasis, all piled right in here. We're doing that because it's just these additive values that we need for parent-offspring resemblance and looking at effects of selection from one generation to the next. These two things are independent, statistically independent, by the regression argument that Joe presented in the first lecture. So if we take the phenotypic means, the means have the same property of additivity that the individual values did. And now if we look at the phenotypic variance of the traits, there's also a set of covariances. So the P, capital P, the phenotypic variance, this is the phenotypic variance of the body count. Here's the phenotypic variance of the tail count. And here is the phenotypic covariance of body and tail counts, right? These you could estimate just from a plot of individuals. Might be small, might be a large sample. So this is a familiar thing. You could see these values popped into a matrix. We're saying that the phenotypic covariance matrix is the sum of two matrices, the additive genetic variance G and the environmental variance E. And here's what those two matrices look like in the two-trait case. And I think I'm going to pop back to our example in the next slide. But we've got, this is the additive genetic variance of the first trait, additive genetic variance of the second trait. Here's the additive genetic covariance of the two traits. The matrix is symmetrical, so these two values are the same. And you can see all three matrices are. Now, if we want to go deep into multivariate space, we want to go to n traits. We just go, I like to think of Star Wars, where you push that lever and you go, woo, and you go into, you just do that, woo, and these matrices evolve to as large a size as you want. Okay? And then we're going, woo, we're going to come back to the two variable case because it won't hurt our heads as much. Questions? Happiness level? Still comfortable? Smile if you're happy. Good. I can't ask. Are you happy at home? Tweet someone in the class. Tell us if you're not and why. Okay, so I didn't have the slide I was thinking. So this element we get from the resemblance between daughters and their mothers in body vertebrae. We get this one from the resemblance matrix plot that we did last time. Daughter's tail plotted against mom's tail. And then these elements are estimated by some average of those two plots. We might weight those two estimates of covariance by something like sample size or something, right? Or we might just take the simple average. There's an alternative way of combining the two estimates to make a single estimate of additive genetic covariance. Now, a little later, we'll talk about where this additive genetic covariance comes from. But right now, we're just going to take it as something we can estimate and have fun with. All right, so so there's another way of thinking about the G matrix, and I find it helpful to think about all of these different genetic and selection parameters in multiple ways. And so I'm going to drive you a little bit crazy because I'm going to show you the same concepts from different points of view. And some of these you may like, and some of you may not like. Our brains work differently, and so you may like this graphical version more than you like the matrix version. But it's also, even though one may be easier to grab, it's worth trying to practice all the different ways because some of them lead you more directly to important conclusions than the others. They aren't equivalent. It's nice to try and practice all of them. Okay, so here's another way of thinking about the G matrix. Now, if you think about a single trait, we pointed out in the last lecture that the genetic variance is just the variance in breeding values, right? The variance in those little X values. So not surprisingly, if we have two traits, we can think of a cloud of additive genetic values in one trait plotted against the other. And so here I've done an imaginary example, and here's a cloud of values. And the fact that there's tilt to this cloud is an indication that there is non-zero additive genetic covariance between these two traits. Why? Well, we're in additive genetic value space. You just have to think about the space that we're in. And what we're going to do is, so this is a representation of this particular G matrix. And we've standardized the traits so that they have a genetic variance of 1. And how would we accomplish that? How would we make that happen? How would we standardize that traits to a value of 1 if we wanted that to be true? Well, if we knew the genetic standard deviation, if we knew the standard deviation of these values on the, we're saying we actually know them, we could divide all the values by that standard deviation, and then the new variable would have a standard deviation and a variance of 1, right? We're going to do a standard deviation like that later. So now, in this new scaling, this is actually equivalent to heritabilities on the main axis. For right now, this is just an example. But what we're saying is we've got a genetic covariance of 0.8, which is quite high. And if we took this plot right here, this is the pictorial representation of that G matrix. And you can, with a little simulation, get it. And so if we calculate the genetic correlation, which is this value divided by the square root of the product of these two, then not surprisingly in this scaled, the correlation is equivalent to the covariances. Did I do that too fast? Okay, so the correlation, the genetic correlation, maybe I've got this in the next slide. This is looking familiar. Yes, there it is right there. All right. So this is a standard equation, and we've just substituted in so the genetic correlation is the covariance divided by the square root of the two genetic variances. It's a standard formula for correlation. Nothing new there. Okay, here's another convention. It's useful to represent the cloud by its principal components. And I can't really teach you principal components in a very satisfactory way, but here is the principal components have different names. So here's the first principal component, also known as the leading or first eigenvector. It's just a new axis fit through the long axis of the cloud under the argument that you're trying to account for finding the direction that accounts for the most variation in the cloud. And then the second principal component, second eigenvector, is fit perpendicular to that first axis. Yeah. The slope? We'll calculate the eigenvector. Let's do it in R. We have a function called eigen. And we'll take we call our matrix G. We'll actually do this, and then we'll go eigen, parent G, and it will give us a little table with the eigenvectors and the eigenvalues, and we can plot them. We'll give you an example of that. And so it's convenient to fit an ellipse to describe this set of axes. And what I've shown here is the 95% confidence ellipse. So if you think of just this axis and go back to think about what the 95%, the upper 95% confidence boundary is, do you remember what the distance is to that edge from the mean? Off the top of your head? What about, off the top of your head? The standard deviation in this direction times 1.96, right? Does that make, so this is plus 1.96 standard deviations that way, minus 1.96 standard deviations this way, and likewise on this axis. Or if you're in R, and you could fit an ellipse by I if you wanted, but in R there are functions that will calculate the 95% confidence ellipse, and bang, it's on there. But then, so you'll see, when Adam does his evolution of the G matrix plots, he doesn't plot an ellipse, he doesn't plot the points, he just plots the eigenvectors and the sort of standardized eigen. So what are the eigenvalues? I didn't tell you about those. That is the variance in this direction and the variance in this direction. This is the eigenvalue for the first trait, the variance in that direction. Here's the eigenvalue, which is the variance in this direction. And so you can plot the 95% confidence limits. I did that in a little weird way, but Joe, use the microphone. Yeah.
I may have missed it if you said it, but another way of thinking about the first eigenvector is it's the line, if you put a line through those points and then project each point perpendicularly onto the line, it's the distribution of those points along that line has the greatest variance. So it's the direction in which you have the most variance is the first principal component. And then the second principal component would be taking out that first principal component. It's the direction in which you have the next most variance, and so on. So
it's good to say it a couple different ways.
Yeah.
It is also, so principal components, eigenvectors, latent roots. There's an older term you may see. Can you think of any others? Principal vectors. But so this isn't like, this isn't something we invented in quantitative genetics. This is a standard tool in multivariate statistics. Okay. Okay, let's look at an example. I sort of alluded to this, but here's our garter snake example. Here's our body plot. Here's our mom tail, daughter's tail plot. Sorry, plot, not count. And what you want to think of is the genetic variance that you estimate from this by regression. And here you can estimate, as I said, the genetic covariance by regression. Again, you're not using the regression slope. You're estimating the covariance from that positive relationship. And then you take these two plots, average them in some ways to estimate that element in the matrix, which is the same here. And then here are the two genetic variances, right? And here are the actual values that we estimated from those data. Now, these are the unstandardized genetic variances and covariance, just for example. And this I'm showing this matrix because if you wanted to use this matrix for something like project the population into the future under selection or drift, you would use this matrix, not a standardized one, not the heritabilities and so forth. We'll see that a couple slides coming up. Okay, so if you go out and estimate, look at the literature on quantitative genetics. Here's a compilation by Tim Massot, who's at the University of South Carolina at Columbia, and Derek Roth, who's at the University of California, Riverside. They were exhaustive up to, what was this, about 1985, something like that, survey of the literature. And they looked at genetic correlations in two kinds of traits. These are morphological traits shown in black histograms. And what you can see, now we have a parameter. So this is our standardized genetic correlation. It's going to vary from 1 to -1, like old correlations do. And you can see that the genetic correlation is predominantly positive, but there's a big tail. But the point is there's a huge range. And I think why isn't it sort of uniformly distributed about sort of balanced distribution centered about 0? And I think it's primarily because people who estimate morphological correlations are doing morphometric studies. So you're measuring body size, and predominantly traits are positively correlated with one another as an expression of the differences in body size. And so that more or less accounts for the shape you get. If you look at life history traits, survivorship, fecundity, and so forth, you get a distribution that has that tendency, but there are quite a few more negative values. And of course, in the life history theory, we're expecting some negative genetic correlations between traits, right? So, and you do see that. All right, so hard to just pick a simple value of a trait to use if you're doing a study of morphological evolution of morphological traits. The more sophisticated packages would use this entire distribution as a prior. We'll talk about that later too. All right, when we had a single trait, we wondered why genetic variance wouldn't disappear over a period of time when we think about the population's quandary of having its variation nibbled away, as Lush said, each generation. We have the same problem when we've got a genetic cloud of points instead of a simple distribution, a bivariate or a multivariate cloud. What maintains that? And so this is Landy's theory for a G matrix now under mutation selection balance. So we've got input of mutation each generation. We get losses by stabilizing selection. We have production of linkage disequilibrium by selection, and its destruction by recombination each generation. But the point is we could equilibrate under these forces, and what Landy shows is it's not unreasonable to expect an equilibration that would produce the kind of values that we see in natural populations. So you can imagine that if you've got a constant force of mutation, a constant force of stabilizing selection, you might equilibrate the whole G matrix at some non-zero values, that its elements would be non-zero. And of course, you could have a more complicated value, as we did before for the univariate case with, before we had migration as an additional source of variation, and then the loss of variation by drift, by finite sampling each generation. Okay, so why is this important to us? Well, it's tremendously useful to imagine that the G matrix might be constant through evolutionary time. And this argument here provides a theoretical framework for supposing that might be true. Now, unfortunately, as we're going to talk about in Adam Jones' lecture, we don't have a really good quantitative theory that tells us about the evolution of the G matrix. And the problem is that if you combine mutation, selection, and finite population size, the actual equations for how the G matrix is changing through time are too complicated to solve analytically. And so you have to resort to simulation. But as we're going to show in our computer exercise this afternoon and in Adam's lecture, which is sort of the lead up to that, under certain conditions you can get nice equilibration of the G matrix. Under other situations, it's going to be unstable. And so the exercise for the afternoon is to get good intuition about when equilibration of a nice stable G matrix is likely, and under what conditions would that be kind of a speculative expectation. This is a good time for a question.
So, I mean, I was just wondering if we necessarily have to assume that the G matrix is equilibrated.
No, we don't. No,
Okay.
it's handy if we can, because then we have simple equations that don't have the G matrix as well as the mean of the genetic value distribution, hence the mean of the phenotypic distribution changing through time. So we could always produce coupled equations, but like I say, the problem is we have equations for how the mean changes through time, but not an equation for how the matrix changes through time. You could do simulations. You could resort to other approaches to try and handle that case, but.
Because I
So
mean,
I'm purposely alluding ahead so you can see. We're going to talk about this in detail in the afternoon lecture. I just want to point forward to this is sort of a simple view of the problem and presents the optimism that we might get equilibration, but of course we might not. And
In the literature of the theory in evolutionary quantitative genetics, it's very commonly assumed that the G matrix is constant because if you assume it, you can do things.
okay.
And one of the most common criticisms, but one which I ignore, is people who stand up at the back of the room and raise their hand and say, but it's been proven that the G matrices are different in different species. And to which you just have to say, fine, what can you do? What can you do with that? How can you use that to make any predictions? And they say, well, we can't make any predictions right now, but if you'll give me another $1 million and let me go back to the lab, I will prove yet again in more detail that the G matrices are not constant, leaving us still stuck. So it's a situation where we wish they were always constant. We act as if they were always constant. But Steve will now tell you in detail that they're not. But sometimes they're close.
under some conditions, they can be approximately constant. So I was talking to Russ Landy. So this is Russ Landy's model. And I was talking to him one time, and we were contemplating the decade of controversy that erupted and still continues after he published this proposition that there might be equilibration of G matrices, so you can consider it a constant. And so huge controversy, papers back and forth, on and on and on. He said, you know, why did I ever say constant? If I just said relatively constant or subject to small fluctuations, we could have avoided 10 or 20 years of back and forth over this. So anyway, but you know, well, anyway, controversies are fun. All right, so now let's take a look at the genetic covariance. We've just talked about it, how it might equilibrate, how we can measure it. What is it? Well, the standard way of thinking about genetic correlation or covariance is to think of it as arising from two sources. And one source is correlational selection. And I've given here a very simple example. This is completely contrived. It's not completely out of left field. There's a family of grasshoppers called the acridids. And they often match their background, and they often come in different colors, and they are coordinated. So here we're thinking about a hypothetical acridid grasshopper in which there's this morph produced by this genotype, capital A allele at the A locus, capital B allele at the B locus, and any allele other of the two alleles at the other locus, and a brown morph that's produced by this genotype. I'm just contriving simple two-locus genotypes to see how we could do this in the simplest possible case. And then we've got these two guys, or gals, in which you've got brown heads and green bodies, and a green head and a brown body. And in our happy and unhappy little world, this is a great morph if you happen to land on green vegetation, be sitting on green vegetation when the bird comes by. This is a great morph if you're sitting out on the barren ground. But this is likely to be more unhappy, right? Because it's unlikely that you're going to have your head on the ground and your body, right? So the point is, and reciprocally here, good morphs, bad morphs with respect to predation. If you think about the effect of that selection generation after generation, it's going to increase the frequency of these two morphs and the corresponding genotypes. And that kind of selection is going to produce a pattern of genetic covariance in the alleles at those two loci. And we're actually talking about the production of linkage disequilibrium at the loci underlying this trait. We wish to discuss, yes? Wait a minute, let's start with Susan in the back row. So, yeah.
All right.
Just for balance.
This may be a very naive question, but this example reminds me of it. What if you don't have stabilizing selection, but instead you have disruptive or directional selection? How does that potentially affect how the gene matrix may or may not change?
It's going to be different, but I'm not going to go into it. So for reasons we're going to talk about later, this is putting it too strong. The smart money is on stabilizing selection as the commonest form of selection out there. And I'm going to show you some evidence for that. But I've got to admit, there are a lot of empirical examples of disruptive selection, almost as many demonstrations of disruptive selection as stabilizing selection. The reason I and a lot of other people prefer stabilizing selection on single traits or multiple traits as the default case is, it certainly helps us explain stasis in the fossil record, which is a very, very strong signal. And so that's why I think in a micro framework, probably we should be, as a default, thinking about stabilizing selection. And this is a version, this is the, we'll talk about the selection description, you know, the algebraic and graphical description of the selection I'm talking about here. But it's a multivariate variety of stabilizing selection. Yeah, comment? Does that help? Yeah. I'm just going to point forward to future lectures when we actually get into selection.
So in those
Felipe will not talk.
In those kinds of genes or genotypes, let's say they are so highly correlated that can we just take them as one for practical purposes?
But for recombination, right? I mean, if they're very, very tightly linked, that might be true. But in general, recombination is going to destroy the non-random patterns of at these two alleles, at these two loci that selection is producing. So.
But I mean, yeah. But then after, I don't know, 1 or 2 generations, that those in that case the recessive genotypes will be just wiped out, right? So
you're an optimist like I am. Yes, you can imagine a world in which, and actually people talk about this as the possibility that such extraordinarily tight linkage evolves that you've got just these alternative genotypes, loci very, very close together, so they're never destroyed by, or recombination is so unlikely you never produce these guys. Could happen. Those are sometimes called super genes, little packed genes that produce alternative morphs. Okay, so that's one way to get, and sometimes you can, when you look at traits, you go, yeah, it's plausible that they might be produced by, the genetic correlation you observe might be produced by linkage disequilibrium arising from this kind of selection. Now let's see, where am I? Oh. That's funny. B. It's like I left out a thought here in this. Okay, so the other way of producing a genetic correlation, I thought I had a slide for this, but maybe I dropped it out or never produced it, is pleiotropy, right? So you can imagine that not all, but some of the genes that affect this coloration, this is going to be multi-locus. Well, let's think of this more plausible for the vertebral case. For the vertebral case, we see a genetic correlation between the body and vertebral count, right? One possibility, and probably the most probable possibility of the two that we're going to consider, is that some, maybe many of the genes are have positive effects or negative effects in parallel on both traits. There's a preponderance of positive and negative effects, and that pleiotropy is what is revealing itself in the genetic covariance we look at. So in general, there are these two explanations, not linkage per se, but linkage disequilibrium and pleiotropy. I still can't believe I didn't have it in there, but anyway, I add it back in. Okay, now we're going to topic 5, and we're going to look at the response of the multivariate mean to selection. Now, with this slide, I just want to produce an argument analogous to the earlier plot we made, right, in which we looked at selection on an offspring parent plot. Okay, so now we're going to plot offspring value for trait 2 against, and on the x-axis, mid-parent value for trait 1, right? So now, by contrivance, this cloud, in its negative inclination, is expressing a negative correlation between the two traits, right? So one way to look at so now we're just going to apply the same argument as before. If we had truncation selection on trait 1 and take the mean value for that set of selected parents, the difference between all potential parents and the selected parents is now S, the selection differential. And if we go up to the line, all parents would produce a phenotype here, and selected parents are going to produce a phenotype, an average offspring value here, and so the response to selection is going to be negative. So now it looks logical to look at the regression, but think how counterintuitive that is. We've actually selected for an increase in one trait, and we get a negative response in the other trait. So this is, you with me? So this is the surprise. This cartoon is just to illustrate the surprises that can be produced by selection when you've got genetic correlations between the traits. And in particular, you can have situations in which the direction of selection is different from the direction of the regression between the two traits. Right? And we've provided, here's a negative correlation, but selection is this. So you get a counterintuitive response to selection. And you can contrive many other cases. So this slide also helps us understand why the animal breeders and plant breeders incorporated this concept of negative genetic correlation into their breeding designs. Because if you're doing a breeding program and you're selecting on, say, some favorable trait, and you get these negative responses in other traits you're interested in because of negative correlations, you need to account for that in your breeding program. You'd want to, in the ideal case, you'd want to take this negative correlation between these two traits into account and design a program of selection, a direction of selection that would produce favorable responses in both traits if you can do it. And this formulation, although it's not so obvious from this slide, the matrix formulation that we're dealing with is capable of that kind of contrivance. And so it was first pioneered by animal breeders, plant breeders, and then adapted by Russlandi and recast to deal with evolutionary problems. But this is the phenomenon of correlated responses to selection being in an unfavorable direction for a breeder. Did you want to comment, or are you just rolling? Okay. Comments? Questions? You sure? All right, let's look at the algebra. So let's look at so when I look at this slide, I realize, this is from my book. And what I do in the book is I deal with selection and responses. I deal with selection, and then I deal with inheritance. And so what I'm introducing in this slide is this thing, beta. Okay, so let's go back and look. This is the breeder's equation. And last time we just presented it like this, right? In the univariate case. So just ignore this. So the change in the mean we showed in the last lecture was the product of the genetic variance divided by the phenotypic variance times the selection differential, right? And so what we did with the breeder's equation, and there's a common convention in which, we take these two terms. If we divide the genetic variance by the phenotypic variance, that's what we call heritability, right? So we could write, in the univariate case, we could write this as h squared s, right? Now, in the multivariate case, this product is no longer a simple number. It's no longer a scalar. It's a matrix of things. And it turns out to be a matrix of partial genetic regressions. And so they're not simple regressions like the heritability. They're these partial genetic regressions. And they're not anything that's easy to think about. But, and this is Russ Landy's result, if we take this product, the product of the inverse of the P matrix and S, that thing turns out to have a simple interpretation, as we're going to talk about for the next few lectures. That thing is going to be a vector that we're going to call beta, and it is a vector of directions of selection acting directly on the trait, accounting for phenotypic correlations between the phenotypic correlations between the traits. And we're going to talk about that in a later lecture.
Steve,
So,
just a word.
A word.
When you gestured at the matrix and said it was partial genetic correlations, that's the product GP inverse.
Right.
The individual G is just the genetic covariances, and
That's
the P
right.
inverse is the inverse of phenotypic
Yeah.
covariances. So the two together are the partial
True.
genetic correlations.
We don't even have a name for this. Steve's matrix. Steve's unuseful matrix of partial I think it's genetic correlations or regressions. I can't remember, and it doesn't matter because nobody ever uses it. It's not a bunch of H squares. Would be nice if it was, but it's not. All right, but we question? That's not even true.
It's not true?
No, it's a mess. Okay.
So the question
I mean,
was...
you could probably contrive situations and when it's. I mean, you could do the whole theory using the selection differential and this instead of G, but it's there's a parallel world in which you could derive everything on that basis using Steve's unuseful matrix. You could make, here's the point I'm making. When we look at the multivariate case, we realize looking back at the univariate case, going down the heritability road was kind of a mistake. If we'd started with the multivariate case, we never would have used the heritability as our measure of inheritance. Are you shocked? So this happens all the time when you go from the univariate case to the multivariate case. Geez, that's not a good way to because the univariate case doesn't generalize to the multivariate case, but the multivariate case generalizes to the univariate case. If that is making sense to you. I don't want to dwell on this too long. This is just sort of an aside, but I'm trying to give you one justification for why we're going to use this formulation for the multivariate response to selection, sometimes called the multivariate Reader's equation. And so now I'm just going to show you the algebra of what happens here. And I'm just going to show later, that this is a vector of direct selection coefficients, more useful than the vector S. And we'll do that in a future lecture, okay? Right now I'm just going to assert, in general, it's useful to collect terms this way and use beta and G, not that formulation. Okay, so if we go down this route, due to Landy about 1979, in the two-trait case, G is this 2x2 matrix we talked about before. Here's beta. I should have used 1 and 2 as subscripts, just a typo. And now we're going to use our first multiplication rule in matrix algebra. If we want to take the product of a matrix and a column vector, an easy way to remember it is rows times columns. So it's going to be this row times that column. Why? It's just like, remember the rules of multiplication? We could come up with, just remember it as a rule. And that rule of multiplication is this times that plus this times that, okay? G11, beta 1, G11, beta 1, plus G12 times beta 2. Here it is here, right? Likewise, that row times this column gives us that result. And now if we look at it, what this is telling us is the response to selection in the first trait has two parts. One part represents the force of direct selection on trait 1 acting through its genetic correlation, its genetic variance. And then there's also an effect of selection on the other trait acting through the genetic covariance, G1-2. Likewise, when we look at the response to selection in trait 2, it's a function of two terms. Direct selection on trait 2 acting through its genetic variance, and a correlated response to selection arising from direct selection on 1 acting through the genetic covariance. Okay, so so what can happen is this can be positive. The variance is always positive. This could be positive, but that could be negative. And so it's possible for this term to be larger than that term, right? And so you can get very counterintuitive responses to selection from the patterns of, especially from negative genetic correlations. Yeah.
Is this on? As someone who teaches linear,
Matthew.
So as someone who teaches linear algebra a lot, something that I see.
Do it, Matthew.
Something. This is on. Okay. Something that I see sometimes in certain applications has a neat interpretation. I don't have enough biological knowledge to know if this is the case here, but when you multiply a matrix times a vector like that, if you do there's another way to think of the multiplication, which is so if you look at the matrix and the vector, take beta 1 times the first column, and then add beta 2 times the second column. And I've seen examples where if you think of matrix multiplication in this way, based on certain models, it has a neat interpretation physically. I don't know.
You mean if you want to take the...
so take beta 1 times the first column and add that to beta 2 times the second column. And that's the...
I'll have to think about that. Well, that is the... Let's see. That would give you this term plus beta 1 times that term, right? Beta 2 times this term.
No, beta 1 times that whole column vector plus beta 2 times the whole second column vector.
Beta 1 times what column vector?
The beta 1 is multiplying the vertical column,
Oh,
the
this
first
vertic-
column, and results in a column.
Well, I guess you could think about it that way. I'll have to...
Yeah.
I'm not facile enough to do it, see what you're doing.
In my experience, it's an alternative way to think of it, and sometimes it has a neat
Sometimes.
interpretation that the other
Yes,
way does not.
yes.
So that's all.
It follows our general rule that there's more than one way to think about all of these things.
Yeah, I just wanted to throw that out there in case you or other people had thought of it that way or had any connections.
Hadn't
That's
thought
all.
of it that way, but
Okay.
I will try.
Just planting the seed.
In the quiet of my room tonight, I will try that. Let's see, what do we want to say? So what I was going to say is, in a truly multivariate case like Wilson's bird of paradise, things just get more complicated, right? And can be more surprising, because now we're going to have a big vector of traits, and we're going to have a very long row of one genetic variance and a whole bunch of genetic covariances, right? So when we get this term, there's going to be one effect of direct selection on the trait, and all of these things are going to be beta 2 times the covariance plus beta 3 times the covariance G1-3, and then all the way out to beta n, the direct force of selection on the nth trait, times the genetic correlation between trait 1 and trait n, right? So you've got all of these possible correlated responses to selection. Pretty easy for them to overwhelm, in theory, the one direct response to selection, right? So the multivariate case tells us that the selection differential probably is not the best way to think about selection, but we're going to make that argument later. All right. So now this hurts that I don't have my computer working so well. But it's okay, because it takes time to bounce out for these animations. But you might try doing that, because I'll have this figure here. So if you click on animation 2, it's going to show this animated. So let's look at what we're doing here. So we're going to illustrate the effect of beta, thought of as a vector, right? So this is beta, in which the first element, beta 1, is positive, but the second element, that would be the direction, this part of the vector, is 0. So it's just a straight line like this, right? And here is the genetic mean, the bivariate genetic mean, right at the center of a circular G matrix. You with me? Okay, now let's think about this as a pull shot, and think of this as the pull cue, and we go like this, and we hit the mean. It'll do a very simple thing. It'll move the mean some distance. And I showed this to someone, and they said, oh, did you just do that graphic? Like, oh, that's the beauty of knowing matrix algebra. We actually calculate the distance. Here's the G matrix. We actually calculate this. We don't just wave our hands. That's, like we have theory to do this. We have theory to play the pool game, right? Okay, so and you can see, can you see that if we do the pull shot in any direction on the G matrix, the mean, and so the whole G matrix is going to move as a cloud of values around the mean, right? And that's what's shown in the, and if we move the arrow in any direction, we would get a response in the same direction as selection. And you could actually calculate that. Now, here's a contrary case in which we've got an elliptical G matrix, and what is this expressing? Tight correlation in what? In what? Felipe? In genetic values, right? Tight genetic correlation. And in fact, you can see that it's, well, you'd have to take 32 divided by 40 would be, what is that? I don't know. Anyway, it's a tight, is that 0.8? At any rate, maybe it's 0.9. No, that would be 0.36. At any rate, 0.8, yeah. I can't even do multiplication when I'm in front on the board. Okay, but the upshot of all of that is if you work through the equation I just presented and do just selection on the single trait, you don't go out the other side. You get an arrow like this, right? And that expresses, remember, this vector has two parts, right? If there's a part this way, that's the direct response to selection. And then there's this part, which is the correlated response to selection, which is almost as great as the direct response, right? And that gives us this sort of weird angle like this. That's the surprise that in the two trait case, that genetic correlation. Now, what if you play the animation, it'll show you different combinations to help you build up your intuition. And then here's a summary slide that shows the case in which we've got this is the same degree of genetic correlation, standardized covariance. And now we're going to try different pull shots, different directions of selection, and we're going to look at the corresponding response of selection. So here's the response that goes with that vector all the way out to the response when to this vector. Now notice a couple of interesting things. When the direction of selection is aligned with the eigenvector, the minor eigenvector or the major eigenvector, the second or the first, you get a response in the same direction as the selection. But otherwise, you do not. And so you have I'm giving you a homework problem. In a single sentence, describe what's going on here. So in a single sentence, describe how the responses to selection different from the angle of the direction of selection. And why am I giving you this homework assignment? Part of this is we're teaching you, did you notice you're learning a foreign language this morning? Unless you're very, like Matthew, very fluent in linear algebra. Most of us have to struggle because we've got these new words. We've got eigenvectors, we've got eigenvalues, we've got directions of selection, directions of response selection. You need to put all of that into a single sentence. That'll be a good exercise, and try and describe this pattern. There's an animation that you might want to play to help. Have I gone over yet? Have I gone over a little bit, have I? I think I'm close to the end here. Oh yeah, this is the end. Okay, so what have we learned? There are really just three big takeaway points. Just as the genetic variance was the key to understanding univariate response to selection, G becomes the key to understanding multivariate resemblance, just as G, the scalar, is the key to understanding univariate resemblance between parents and offspring. I jumped ahead of myself. And it's because of that, because of the because that plot takes us from one generation to the next, that kind of resemblance, the G matrix is the key to understanding multivariate response to selection. It is the matrix that takes multivariate selection across generations. The thing that makes it non-intuitive is that the G matrix can induce correlated responses to selection that may or may not, but often are non-intuitive. And so it takes a little practice either with algebra, linear algebra, or cartoons, or whatever helps you to make those responses intuitive. Here are some references. And there we are. Okay, so let me just look at our schedule and see where we are. As usual, I went over, but if we're supposed to have discussion until 10:15, so we have a magnificent 5 minutes, and I apologize. We had a few questions, so we had a little melding of discussion. But now I'd like to hear your questions, if you please. And Tyler is going to be the microphone person. Let's see, who could I call on? Okay, Dave.
I just have a geeky algebra question. So when you're deriving...
I like geeky algebra questions.
Excellent.
Yeah.
I have one then. So in deriving the response to selection, so the change in Z1, so if you go back to your matrix notation...
Oops, that one?
I guess it's very non-intuitive that the main diagonal are not, so G11 and G22 are not the heritabilities.
Of the G matrix?
Correct.
Yeah.
But I guess if you were to go out in the field and take a univariate approach from the beginning and just measure heritability and selection on a trait. So that heritability and selection would not equal to G11 times beta 1.
Say it again.
So if you were to first take just a univariate approach and measure heritability on a trait, selection on the trait, and also evolutionary change in that trait, essentially the Z1, G11, and beta 1. So you said that...
But how are you going to measure selection? That's the trick.
Well, didn't you teach us how to do that yesterday?
No, we're going to do that later today.
Well, I thought you could just take from a univariate perspective, you could just look at the difference from the parent population, the mean in the parent population minus the mean of the selected population. Isn't that the selection differential?
Yeah, it is. But so here, this is one of the heartbreaks, the multivariate heartbreaks. No, you've got to take, so.
Can I clarify? I think you guys are talking past each other. Can I see your microphone? Because this is something I deal with in my quantitative genetics class.
Please.
So I think what he's getting at is that when you're just thinking about this in terms of an algebraic construct, if you're just thinking about a single trait, it's absolutely fine when you're thinking about a single trait to break the heritability into the additive component. So you have V sub A times S times V sub P minus 1, right? You can set for us. This is what you're asking, I think. Yeah. So you can absolutely do it that way.
You want to do a thought experiment.
Yeah. For a thought experiment, you can do it that way, and then if you want to make in a univariate case to make it look like that. More like this equation here, you keep your V sub A separate and write S divided by V sub P, okay? And then now it's taking on the form that you're talking about. And that's absolutely fine to do it, to think about it that way. And in fact, I encourage, when I teach univariate response to selection, the breeder's equation, I do it the normal way with heritability, and then I say heritability is going to be a liability later on when we start talking about the multivariate case, so just humor me and let's just rearrange the equation algebraically to break out the V sub A and divide S by V sub P, and just think about it, just and people go, okay, well, you want to do that, that's fine. It makes sense to them algebraically. Then when we get here, they're like, oh, I see why you did that.
Yeah.
So it's absolutely fine to think about it that way. And I think that's what you're wanting to do, if I'm correct. So yeah, absolutely think about the univariate case that way. And then when you get here, it's like, oh, okay, we're just adding an additional trait, but the terms are still the same. We have a response to selection is equal to a variance component, the V sub A or the G matrix, times some sort of selection component, which is either a beta, in multivariate case, beta, which is a vector, or S divided by V sub P if you're just thinking about a univariate case. Yeah.
But when you do that, you're making two assumptions, right? You're saying selection is, my thought experiment is selection's acting on a single trait, and it's not genetically correlated with, it's not phenotypically correlated with any other trait.
Yeah,
right?
let me just add, along the same lines. Yeah, you can go out and take a case which in reality is one of these two character correlated cases, ignore the second character, measure just the first character, measure the selection on it, get the heritability, get the selection response, and you know what? You won't get the right number. The selection response won't be that because there's also selection going on the other trait, and there are indirect correlations. But you can do it, but your beautiful isolated world will be shattered when you actually look at what the response to the selection is.
Or you might get lucky and it,